So today we have Dr. Leon Zhao. He is a chemist and biomedical engineer with vast expertise ranging from protein engineering to neuroscience, chemistry, and optics. He is the main inventor of the genetically encoded calcium ion indicators known as the Gecko family, and the genetically encoded voltage indicators named COSAR. And so these have been indispensable tools in thousands of neurobiology labs across the globe. He has also been a key inventor of innovative imaging tools grounded on expansion microscopy, enabling nanoscale imaging of various biological specimens and clinical biopsies. So he's at Carnegie Mellon, and he spearheads a vibrant research group with the mission to develop and apply new tools for establishing an ultra-high resolution biomolecule atlas of the brain and other complex biological systems. Currently, his team is on the cusp of developing an array of tools that allow for precise spatial profiling of biomarkers in human and mouse tissue sections at nanoscale resolution. They're also pioneering computational tools, including deep learning, to derive insights from nanoscale tissue imaging of patients for novel diagnostic tests. He has been involved in developing next-generation nanofabrication technology for biomedical applications, epitomizing his multidisciplinary and collaborative approach to addressing formidable challenges in biology, pathology, and medicine, along a host of collaborators from various labs in the US and abroad. So his talk today will be on Magnify, so the next generation expansion microscopy, with the universal molecule anchoring strategy and its applications and beyond. So thank you, Leon, for being here. Please take it away.
Sounds good. Thank you so much for the comprehensive introduction, Natalie, and I'm also grateful for the invitation from IMS. And can you folks see the screen? Good, awesome. Great, so since Natalie already gave a very comprehensive introduction about our research group, I'm actually happy to share the recent progress that we make so far towards the goal of establishing a biomolecule atlas for any given humans and other animals in the normal physiologic and disease states. And I just a disclaimer, I'm also a scientific co-founder and advisor for a startup called Magnify Bioscience. So since the audience is expansion microscopy user groups, I'm not going to talk about like what's the motivation behind developing this expansion microscopy. I'm going to quickly just go through this this concept, right? I mean, we are assuming that Switzerland is the Earth, and then you try to use a conventional microscope to see a specific cell type. For example, in this case, it's building. You know, the building that I'm working on, it's Mellon Institute. Due to the diffraction limits, it's really hard for you to see the details about these buildings. If you can actually figure out a way to expand it in 3 dimensions and then using same microscope, then now you will be able to see the expanded Mellon Institute and be able to see everything, all the details with much higher resolution. That's the metaphor. And quickly, just to recap, right, what was the traditional way to do expansion in microscopic level? What happened? So you have to have an anchoring reagent, right? You try to, it's a small chemical handle. You try to modify the biomolecule of interest. Usually it's very specific. You have to pick and choose. And after that, you modify the tissue and then you put them in a gelatin solution, right, containing all the building blocks. And you trigger the polymerization reaction and form this polymer chain. And those modified biomolecule with the handles now be able to covalently link to this growing polymer chain. So imagine you kind of are drawing a super dense polymer chain mesh, and where all the biomolecule of choice are actually now covalently linked to the gel network. And now if you try to treat the sample, we call this homogenization. You can apply enzyme, you can digest all the endogenous protein that hold the tissue together, or you can try to heat it up and denature them. Then you will be able to actually enlarge the sample. The reason is the hydrogel itself is water soluble, right? Just like commonly used baby diaper material. And we of course we know that you have to do quantification very carefully, like for different methods of expansion microscopy, you need to actually carefully do this validation, especially if you try to apply this to different new tissue type, right? That are not characterized or reported before. And so you need to use this kind of quantitative tools. Of course, nowadays there are others methods, try to characterize the distortion. Actually, I saw multiple publications recently. But this still becomes the most common way to do it. Now, my role for this development of expansion microscopy world is a little bit different than the majority of them. I mean, my motivation is actually try to bring this expansion microscopy to clinics, to pathology. That's my ultimate goal. So during my postdoc work with Professor F. Boylan, who actually is first published the science expansion microscopy paper. So I worked with him to develop the first generation of pathology optimized expansion microscopy. This is called expansion pathology or expansion path. So compared to the original version of expansion microscopy, is that we developed some aggressive protease K digestion that are capable of processing all the non-brain human sample. So here, just give you one example. For example, this is a human kidney section, FFPE. We remove the paraffin, and after that, you treat this sample with the expansion pathology procedure, and then use this enhanced protease K digestion. You will be able to expand the sample as shown on the right. The good thing about this is that we can also use this kind of method to process H&E, for example, which is one of the classic way to stain the human tissues and be able to examine by the pathologist. Now we prove that we can actually expand this and then be able to demonstrate you can add additional layer of information with nanoscale resolution. You can do immunostain, for example. You can also do DNA fish. This is just some of the showcase. I'm going to quickly go through it. And it's a DNA fish on HER2 positive and negative breast cancer tissues. As you can see, we also actually do immunostaining to show that the expression of the HER2 are really correlated with the amplification of the HER2 gene. And of course, we can do this on many different types of human tissues. Here, just list 8 of them. They represent the common organ that have cancer. So we also show normal and cancer. It's a little bit busy slide, but then basically that's the whole message to show you that we can use expansion pathology to expand all kinds of different tissues. And of course, we try to demonstrate the utility of this kind of method to explain why we want to do that, right? So we picked 2 case study. One is kidney. So kidney has a lot of needs for nanoscale imaging, and actually clinically they have to use electron microscopy to do certain diagnosis, maybe like 30–40% of the disease. So you can see here, have in the middle it's a scanning electron microscopy images of kidney podocyte. You can see the very complex processes right under this microscopy images. This is called podocyte full processes. If you do a cross section, just like the EM does, the TM does, and you will see the periodic structure, right, like this, and that's the normal state. And if it becomes continuous, that will becomes a disease. It's called minimum change disease. Of course, there's other disease, kidney disease will associate with this kind of change. As the name suggests, the feature size is smaller than the diffraction limit. Therefore, using optical microscope cannot see. The question is whether you will be able to apply this expansion and be able to see that using conventional optical microscope. Right here, just shows you the result. You have these electron microscope images of the corresponding podocyte full processes, and then for the similar tissue, we expand it. And then you will be able to see that we stain the full processes with the actinin 4 And you can see this periodic structure similar to that shown in the electron microscopy images. And in the case of kidney minimal change disease, in the EM, it becomes continuous. As expected, you expand the same thing from those patients, you will see the processes become continuous, similar to the EM. We even do a single-blinded experiment where we train a bunch of people, some normal regular scientists, and some of them are pathologists, and let them look at the expansion pathology images as a sample, as well as the fluorescent, and then try to ask them to tell a group of unknown images. And it turns out if we show them the conventional fluorescent images, they have hard time telling the difference. And while if you show the expanded one, they have accuracy of more than 91%. Second demo is on the breast cancer. In this particular case, it's not quite the cancer yet. It's a precancerous lesion. We know that lots of women will have a high chance to get breast cancer throughout their time. The problem here is for the early diagnosis, because if they want to diagnose, like they need to do the mammogram and after that they do the biopsy, sometimes they come out with some borderline cases. It's called atypia. The problem is atypia looks very like benign without atypia. So it turns out that the pathologists will have a hard time telling the difference. If you have 100 pathologists, only 50% agree. That's a problem. The reason is depending on different criteria, the diagnosis with atypia, the medical oncologist might consider excise the rest. So if you either underdiagnosis or overdiagnosis will become a problem. Now, our rationale is what caused that? One is, of course, it's a qualitative criteria, right? Everyone has different opinions, slightly, even though you have a textbook. Second is they use blind field imaging, which has a very limited resolution, and therefore they only get a limited information out of that. So in order to overcome that, we collaborate with Professor Dr. Andy Beck, and we try to actually apply computational models, try to process these H&E images as well as expansion images. We want to do a side-by-side comparison. First, we look at the segmentation. As you can see here, with the expansion, you get much better efficiency to detect individual nuclei on the images, while if you do the same on the H&E, you get much larger false positive rates, as indicated by the red, right? You compare to the expert annotation. So even just the segmentation task alone, expansion, the improved resolution really helps. And then we try to segment, and after that, you try to train a machine learning model. In this case, we don't do those fancy deep learning model here, just a linear regression, but we do a binary comparison. As you can see here, I highlight the two in the middle, the two groups in the middle, and one of the group is comparing a benign without atypia versus atypia. And as you can see that even with AI, which shown as a purple dots, you only get like 70% accuracy, while if you increase the resolution by 4 times, in this case, you can get a much higher accuracy, thanks to the features that enabled by the expansion, which is great. Now, after that, I become independent, and I start to actually try to continue to improve this kind of tissue expansion for pathology application. One of the first things that stand out is expansion pathology actually lasts for 2 days. It's a 2 days process. It's not fast enough for the clinical application. So we developed this rapid expansion pathologies that only costs you like 8 hours. This is one example. We take the HIV containing lymph node from patients, and then we use only like less than 8 hours to stain and then process the tissue, and then got the images. Now, all the stuff that I talk about all fall within the traditional expansion microscopy regime, right? What's the problem? The problem is that most of them are only expanded 4 times. So the resolution, if you calculate a little bit, even use the best objective, you can only get like about 17 nanometers. Now, the second problem is I heard, maybe you folks also share the same pain, is that the handling of the gel is quite delicate. You use a brush. I guess people use brush to handle, or you can use other stuff. Brush still becomes the most flexible one, but I mean, it still breaks gels. And sometimes they try to wash the gel, and then you suck it up right by the pipette. I think everyone probably has some similar experience. Not to mention, if you want to image a certain molecule, the problem is you have to pick, right? If you do RNA, you cannot use a protein retention expansion microscopy because that did not anchor the RNA. You have to pick expansion fish, for example. And most of the methods are actually use enzyme, right? So you digest everything actually. You're essentially looking at an empty gel with anchor antibodies. Not to mention, except expansion pathologies, everything else is pretty limited in terms of expanding the tissue type. Now, so this is just a review that we summarize what's going on with all the traditional expansion microscopy. You always have an anchor step independently, and then you gel. After that, you homogenize, and then you expand, which is fine. The thing is, the anchoring step are really like, you have to pick and choose. And the homogenization always use enzyme. So we try to modify these two steps, right? First, I don't think actually you need the anchoring a step independently. That actually limits many applications, because like if you use some very sensitive reagent, there's a lot of thick tissue you cannot do. And second is like if you use like protein digestion, then you basically miss the opportunity for highly multiplex imaging later. And also you cannot do post-expansion immunostaining, of course. So to overcome those challenges, we developed this Magnify. We also try to change the gel formula so that you can expand actually 11-fold. We have a universal molecular anchor. It's called methylcholine. Methylcholine is kind of like a fixative. It's actually used in the plant science for decades as a fixative. The good thing about this guy is it actually broadly reacts with all the biomolecules, while it still has the double bond to be able to serve as a monomer to participate into the polymerization reaction. So because we changed the gel formula, so we expand 11-fold, but we still keep in mind that the people will complain about like delicate gel handling, right? So we also enhance the gel. Here just shows us some example. This is an 11-fold expanded gel. It's magnified gel. We try to use the same formula to generate geometry like a half cylinder, so that you let the round side sitting on the ground. What happens is if the gel are not sturdy enough, it will deform, right? Because you expand 11-fold. That means that 99.9% or even more space are actually filled with water. It's super heavy compared to its own weight. But you can see here, like Magnify can actually hold itself even after full expansion, while the competing 10x expansion actually collapses itself. And because we use a universal anchoring, we have to characterize a little bit like what exactly retain, right? So we test the protein first. As you can see, it's a quite busy graph, but basically the take-home message is that we test the protein retention efficiency across different types of tissue. And it turns out that whether or not you use enzyme or you try to heat it up, methylcholine tend to retain more proteins compared to the traditional protein retention microscopy reagent. It's called ACAS. Because you retain all the proteins, and also we use a heat denaturation method to homogenize the sample, now we'll be able to actually do some sort of a pan stain, right? Just like the pan ESM or others use. You can stain with an NHS ester. In this case, we stain the mouse brain, and then you'll actually be able to find those synapses just like the EM does, and you'll be able to see the presynaptic density projection, presynaptic dense projection. The panel 1, 2, 3 are actually magnified images. I try to make it fake images just to look similar to the electron microscopy. And of course, with this 11-fold expansion, you will be able to get a much more accurate measurement between this presynaptic and postsynaptic markers. So this is just one of the showcase that shown that you can do something like that, across different brain regions. what's interesting is that because it's a universal anchor, it turns out you can actually anchor endogenous lipids. Previously, there are multiple attempts to retain the lipids in the expansion microscopy examples. The problem is they always like add the third party, kind of like quote unquote third party lipid, right? It's a modified lipid that with a handle to cooperate into the polymer. In this case, it's actually endogenous. So we found that the retention are pretty efficient. Even you treat the sample in heat, and for 24 hours, you still get like more than 50% of the lipid cap. With that, you will be able to actually stain the sample even after you expand, right? You can use traditional lipid dye to stain. In this case, we use DID. I'm just showcase one of the example, right? This is a synapse. You can see that in my cursors, like this is actually a presynaptic compartment with this range, and you have all the yellow, the synaptic vesicles, and also like this little purple thing is actually the mitochondria. You can see even the crystal structure. Of course, like with that, you can also use Magnify to actually treat the transgenic mice. So here it's example showing this mouse brain from transgenic mice where the dopaminergic neuron are actually labeled with a TB tomato. So we can actually expand this samples in Magnify gel and then be able to see this native fluorescent signals from the TB tomato. You can also stain different kinds of molecule, right? This is fluorescent protein. This is DNA, right? This is lipid, and this is carbohydrate. So it's a lectin, right? It's a tomato lectin that bind to those specific sugars. That highlights the blood vessel. Because you keep the lipid, so we can do something interesting. For example, you can actually image the secretory extracellular vesicles. In this case, we do that on human stem cell derived lung organoid. The G and the J shows the panel of electron microscopy images of that mucus cells that's secreting this kind of vesicles. Now, the rest of them are actually magnified images. So we expand them, we stain with protein as a green and then the lipid as a red. And you can see that we can image actually 3-dimension compared to the electron microscopy with ease, right? You can do the 3D imaging. You can also pick one of that. For example, this is one of the guys that I picked that you can actually dissect optically across this vesicle. And it turns out this kind of extracellular vesicle have a very complex structure. It's not just a bubble. You have complicated bubbles inside, right? And of course, we mentioned that we can do lipid, proteins. You have to also test nucleic acids. So we do this dual DNA RNA fish just to showcase that we actually can model. Now, people will worry about distortion. So in this case, we also show that Magnify, just like other well-validated expansion microscopy methods, has 3 to 4% distortion. We also do this in a more challenging way, in the sense that most of the other expansion microscopy methods, they characterize that on culture cell, which is easy. But this one is on the FFPE tissues with heavy formalin fix. As you can see that we actually, did not cause any much more distortions. It's also about 3 to 4% distortion rate. And you can actually apply Magnify to a broad range of tissue, just like expansion pathology does, except that if it expands bigger, it actually anchor all kinds of biomolecule inside the gel. So you can do post-expansion immunostain and other stain. And because another good thing about it is post-expansion staining offers lots of advantage in terms of signal strengths. And with that, you can even apply some of the trick. For example, there's super resolution optical fluctuation imaging. It's kind of an approach to extract additional spatial information from a time course images of the same Z set, right? So if you acquire, in this case, we acquire 50 frames or 100 frames of the same thing, same Z, you will be able to extract additional information. So 11-fold expansion or 10-fold expansion can give you about 20 to 30 nanometers resolution, depending on which objective you use. Now, with SOFI, we are able to actually boost the resolution to 15 nanometers. Now, with that, you can actually get to the level to see this kind of cilia structure. So this is a mobile cilia showing here. On the left, it's electron microscopy images. On the right, it's in the middle is the Magnify images of the similar stuff. And you can see that the diameters, everything looks similar. You can actually see this 9-fold symmetric of the microtubule bundles, as well as the singlet inside. The good thing about that compared to electron microscopy, again, is that the 3-dimensional capability, right, as shown here. And we even go ahead and image the whole ciliary machine from all the way from the root, which is the basal body, all the way to the whole cilia, right? So you can actually image the whole machinery using this approach, Magnify SOFI. And of course, we try to do some demos here that related to disease. So we treat the human lung organoid with the drug Taxol that will damage the cilia structure because it hinder the growth of the tubulin. Now, we also try to get a human organoid derived from a patient that with a mutation, they with a PCD, that basically they're born with the defective cilia. So we try to characterize the normal looking and the abnormal looking cilia among those groups, right? Normal, drug-treated group, as well as PCD. We also compare the side by side with electron microscopy. I show this complicated graph, but basically the take-home message is essentially you cannot tell the difference by looking at this graph whether or not the group are measured by Magnify versus measured by electron microscopy in terms of like error bar and all that. Basically, they have similar characteristics. That's the take-home message. That means that this Magnify SOFI has the capability of detect ciliopathy with similar performance as electron microscopy. Now, we check almost all the box, right? So we have Magnify. You can image different kinds of level, right? You can image the whole tissue level. You can zoom into the cellular, subcellular, which is something that even confocal can do. But then you can actually go down to the suborganal level, right, which is about 20 nanometers. And as you can see here, this is a kidney process, full processes again, but you show the high ultrastructure context as well as protein markers and carbohydrate markers, right? So now you actually check all the box that I mentioned, you get better resolution, you get much more robust gel handling, you can actually universally anchor all the biomolecule inside the gel, and you can do all sorts of interesting stuff later. So for example, you can, in this case, right, because you keep the biomolecule inside the gel. So think about it, then you think, actually, maybe you will be able to do label-free imaging, right? So to do that, you think about some chemical vibrational imaging methods. Here, we team up with Professor Wei-Ming Group in Columbia University, who is expert in stimulated Raman scattering microscopy. So we try to combine these two technologies together. And then it turns out that they can detect the biomolecule sitting in the magnified gel. Just showing you, for example, proteins, they can detect lipids. Of course, if you try to apply the antibody that with a Raman dyes, you will be able to detect that as well. But because the expansion, you actually improve the resolution of this stimulated Raman scattering. And if you combine with fluorescent dye and as well as a Raman dye, you will be able to even achieve, high prax imaging in one round. Right here to show you like this is 8-plex fluorescent Raman imagings of a synapse. And then you were able to do the similar quantification, just like what I shown before with Magnify only. But here you can do much more numbers of colors. Now, the thing is, even Magnify can expand a broad range of tissues. There's one type of tissue cannot expand, which is a pathogen-containing tissue, because it has its own challenge. If you look at those pathogens, setting aside virus, the rest of them actually has cell walls, right? For instance, fungus, gram-positive, gram-negative. The problem is they have different kinds of cell wall. It's a unique problem for each type of pathogen. It turns out that my student Sherry found a way that to actually universally treat those samples. So it turns out that there's a cocktail that we found with a cocktail of enzyme combined with Magnify process. You need both, and then you will be able to actually expand this kind of pathogen or the pathogen-containing tissues. So we characterize that using this new approach. We call it Micro-Magnify, stands for Microbiology Optimized Magnify. So we call Micro-Magnify. You can expand bacteria, fungus, or even infected cells. We characterize that distortion, and they are all, about 3-4%. And you can do something interesting now, because you can actually now process infected tissues, right? This is a mouse tongue infected by the fungus C. albicans. You can see those filament or purple filaments on the surface. Right? I show you here, and then you can expand this sample and then stain with different kinds of stain, and then be able to actually, get an additional layer of information out of this. And you will be able to resolve very densely packed biofilm of this fungus. We all know that like those C. albicans fungus biofilm are super dense. It's very hard. You have to do the clearing. Even that you might not have good luck to that penetrate very deep. In this case, because you expand many times, so that everything becomes clear. Now you can actually resolve individual cells in a densely packed biofilm. You will even be able to like detect some of the organelle, for example, like this purple protein stain that actually highlight the mitochondria. Of course, you can also expand the bacteria and be able to examine some of the outer structure. For example, here we showcase septin ring. It's a structure that only show up like during the division. We also try to challenge the infected tissue. So we collaborate with Will's Eyes hospitals from Philly. They send us one of the most challenging tissue to expand, which is a human cornea. Believe it or not, this is much harder to expand compared to muscles. So because it has so much collagen inside, it's really tough. So we managed to expand them using Micro-Magnify, and then be able to actually image the pathogens in those infected sites. And as you can see here, like we showcase fungus, we showcase different kinds of pathogen. You have like this, even an ammonia, an amoeba or microbacteria, right, of all kinds. Of course, not to mention gram-positive, gram-negative pathogen in the context of the tissue. Because all the biomolecules are preserved, so you were able to actually do high-plex imaging with these iterative rounds of immunostaining. For example, here we did 3 rounds and be able to stain actual image like 10 different colors. And of the Staphylococcus aureus infected hex cell in this case. Sorry, not hex cell, U2O2 cells. And you see those little green red dots is a bacteria, and then you have all kinds of different markers that are labeled. Some of them proteins, some of them are DNAs. With that, you think about it, because you expand several times bigger, the pixel size effectively becomes smaller. So you can actually use these tools to perform a colocalization analysis with much higher fidelity, right? By analyzing the overlapping pixels, you are able to actually review the picture of how the protein might interact with each other. So this is one of the demos, right, to show how these 10 markers potentially interact with each other by proximity in different conditions under the toxin A, or the mutated non-toxic toxin. And you will be able to see what kind of protein pair actually stand out during this process. And then be able to analyze the actual interaction. Actually, this part has been covered by Tom and Skillman previously. I'm going to talk about that a little bit in the context of our paper. So we now have a complex data set, right? What to do with it? So we use this kind of virtual reality, try to give a real sense of the data, right? We try to feel it instead of staring at the fake 3D in a 2D display in this case. So we try to use this virtual reality. We collaborate with Tom on this, build tools. It's a collaborative tool. So that means that it's like a chat room. You can actually wear the virtual reality gear in different places in the world, as long as you figure out the right name of the chat room, so you can log in and then be able to share the view of everyone else in the room. Some features including you can actually dissect multiple channels, right? You can actually have these so-called adjustable excluder. The purpose for that is, without this, if you walk into the you try to zoom in and then try to drag the data in front of your face, the problem is that all the data overwhelm you. You cannot see anything, right? So, because you got blocked by the closest data point. So the excluder serves to exclude all the pixels that are too close to you, so that you will be able to see the interior of the data if you try to stick your head into the data volume. So basically, that is some of the tools that we developed so far. We still continue to develop them. Our ultimate goal is to extend all this pipeline to whole organs, thick tissues of all kinds. We try to combine the multiplex as well, so that you will be able to establish the atlas as we mentioned before. The thing about this power, right? Because you do the multiplexing and then you have a co-localization analysis. You can actually generate a very complex, interaction network, right, among different kinds of proteins. That's our vision, right, for the near future, hopefully. As I mentioned, I was still actually a spin-off company, just try to disseminate the technology. One of the motivation is there's just so many different inquiries about Magnify. People, have sometimes have challenge to figure out the source of all the components. So we try to help them to put everything together. And also we also have some other features slides that can highlight the power of Magnify. And this is just one of the website and the icon. Not try to advertise, but I just want to disclose that. And some other interesting stuff is you can actually reverse the expansion process. So in this case, you will expand the sample, and then you actually can apply a phantom second laser to modify the gel, but you can print pattern on it. And it turns out that when you shrink it, you can actually induce some interesting self-assembly process that can apply to different kinds of materials. As we show here, we show 12 different kinds of material that can actually deposit on the printed area. And therefore, we come up with a nano fabrication method that can 10 times better than the resolution of the current best approach, because the shrinking process. I just want to mention this a little bit. It's a quite interesting direction to go. With that, I just want to thank all the funding resources and also all the amazing collaborators, as well as our excellent group members. I'm open to all the questions, and of course, we also are happy to discuss if you have any synergy or collaboration projects. Thank you.
Thank you. That was a really, really good presentation, and the pictures were amazing. Like I've been doing the ProXM, I guess, protocol, and then like a big issue of mine is signal retention. So, you know, seeing your pictures, I'm really jealous. I wish I could have that much signal in my images.
Well, I mean, you will, right? I mean, we have many different ways to help you to do that. I mean,
yes,
because one of the goals is to make this kind of a new expansion process just as easy as Western blot. That's the whole goal, right? To do that. That's one of the mission of our lab.
that's great. If anyone has any questions, feel free to raise your hand or unmute yourself as well. You can type it in the chat and I can read it out loud.
Would it help to maybe stop screen sharing, and then that way we can see everybody in the audience too?
Absolutely. Thank you.
Awesome. Thank you. That was an amazing presentation. Yeah.
Thank you. Yeah, feel free to you folks like Chairman if you have any questions or curious or anything. Yeah, George.
Looks like George has his hand up. Did you want to take yourself off mic and ask George?
Yeah. Unfortunately, I had to step out in the middle of the presentation from a fire drill or fire alarm.
Oh, no.
So hopefully this question wasn't already addressed. In my own expansion explorations, I have started to work with Magnify, and it's been very promising. I've just had like kind of 2 sample preparations so far, so very early stages. But with other methodology, the T-Rex, I've been having a lot of issues with sample flatness in 3D. Have you ever encountered this? And have you ever solved it? Like basically, I have very good XY resolution, but in Z I see curvature in my samples.
What kind of sample you have? Is it freshly fixed samples or?
Yeah, this is primarily here in cell culture, but I've also started working with DFA fixed tissue section. And I've seen this curvature in those types of things.
So I think one of the things you can look for is the accelerator, as well as the gelling temperature. I think some of the issue that you mentioned, actually we encountered it during the development of the method, especially for cell culture. I would suggest that you try to increase the TAMAT concentration, maybe ideally 1 to 100 dilution, and then be able, and also like maybe increase the temperature a little bit and for the gelling so that it will set quickly. Doing that, you will suppress that wiggle issue. Is there a wiggle? Like looks like weird, like the cell have some wiggle and ups and downs.
Yeah, it seems more like the actual gel seems to be flat on the cover slip, but when I look at individual cells, I have this high concave nature where the center of the cell is a bit higher off from the surface and the edges are closer to the surface. And I've also seen a similar behavior with sections where it's a very large scale concave curvature.
I see. So you mean like the gel is curved, it's not flat.
Yeah, so the gel seems to have a curve to it. I haven't fully characterized this, so I'll have to look closely at that.
Yeah, I mean, it would be great if you can show us the images. I mean, like the wriggle problem. I mean, we encounter the wriggle problems more often than this kind of flatness, because usually if the gel are not flat, one of the most likely reason is the sample are under homogenized. So you did not hit enough time to homogenize the sample. Once the sample become like fully homogenized, usually the mechanics stress will disappear. So that it will become flat, right? Because you have one side has a sample, another side has did not. And then the gel forces it to bend towards the tissue side or whatever sample size. So if you homogenize that part, then it will become flat.
Okay, great.
So what system are you imaging your samples with? And is it fully submerged, or are you getting a meniscus that's forcing it down?
Are fully submerged. I do a polyel lysine treatment of the glass and then submerge them afterwards, and they do stay adhered to the glass. And I do a combination of spinning disc and lattice light sheet to get higher. Higher resolution. Yeah, thank you. I'll definitely look forward to seeing the recording to see the parts that I missed. Thank you for the presentation.
Okay. Yeah, feel free to contact us if you still have troubles.
I have kind of a general question. Just you've shown a lot of beautiful images, beautiful staining, and several different techniques. How straightforward is it for other people to implement these? And if I just from the last question, you can tell it's very detail oriented. Do you offer courses, workshops, exchange of experience visits in your lab, etc.?
Yeah, so the ease to use is one of the features we try to pursue, right? But of course, like, while our lab are very happy to try to address all the inquiries. Actually, there are many inquiries we receive. That's one of the reasons why my students try to just get a spin off a company, try to develop this kind of thing, the kit, so that can solve, like, basically hear all the voice and then try to solve all of them within the kit. I think the protocol, we try to make it very simple, and it's as simple as that. We actually have my son, 9-year-old, try to do that and success. Just that simple. There's a question in the chat, I think. Yeah, so tubular is one of the things that we actually spend time on. If you look at some of the supplemental images, you will see that we actually try to characterize the tubulin very carefully. Because we do the post-expansion staining, the lineage error due to the label size of the antibodies become much smaller. So we can actually get 45 nanometers, 45 nanometers. Yeah, we can get 45 nanometers width for the microtubulin, for the microtube, which is very close to the EM result. We can even do that on the organoid that with tubulin stain. Is that your question? Awesome.
They
So
said
I
you
have-
got it. Yes.
I have a quick question. So you said that the kit that you're making, it's going to be 10 to 11 fold. It's about 10 to 11 fold expansion. Is it always fixed at 11 fold? Like, what if I don't want my sample to expand 11 fold because it's just going to get too big? Like, is there an option?
Yeah, I realize that. Like, so depending on what you want, right? Actually, another good thing about Magnify is if you put them in PBS, it expands as big as the fully expanded original version of expansion microscopy methods. So it's about 4 times.
Yep.
So it's very good because that means that you can treat, or basically most of the labeling reagent works in that range. Right? So you can do all the stuff, like still enjoy the 4 times expansion. Actually, even us, we do 4 times expansion all the time. Let them sit in the PBS. So actually, that's one of the huge advantages. If you have a regular expansion, even you keep the molecules, like you can only get like 2 times, 2.5x roughly in the PBS. If you try to expand it and then with the immunostain post expansion, the problem is they will detach in the DI water. So in this case, you don't have the problem. You still get like 67 nanometers resolution.
Thank you.
Thanks.
So I think that takes us to our time. If nobody else has any more questions right now, I guess we can end the meeting there. Thank you so much for coming and sharing your work and all those pretty pictures in the morning.
Thank you.
I guess if
Just a
anybody has-
reminder, I was just gonna have a reminder, it'll be recorded and put on the RMS YouTube channel. So we'll, if you have colleagues who might be interested in the work as well, it'll be there for viewing. So thanks.
Yeah, thank you so much, and feel free to contact us if you have any question or any potential collaborative projects. As you can see, we collaborate, open to collaboration all the time, and yeah, I'm looking forward to actually have a venture with you folks.